Okay, so planets for today. Again, we go back to like the major topics that we want to cover. Okay, is we want to be able to hammer out, obviously, the difference between prokaryotic, eukaryotic organisms in terms of DNA, right? And then we want to talk about the structure of DNA versus RNA. Okay, now the first little bit on here has a lot to do with history. I'm going to bypass the history. Okay, if you want to watch this, actually, if you go into Bozeman Science, I took these slides. They come directly from Bozeman Science. And he goes through and he talks about the history. And I'll address a little bit of it just because I think there's some important things that you guys need to understand as far as kind of the founders, the people that actually found, the DNA molecule itself and how it's not exactly true because they had a lot of help. We'll talk about that a little bit. But the first little bit that we just kind of talk about, just to spark interest in this unit, is kind of where we are going, is that ultimately we're going to end up with gene regulation. Okay. And a lot of our labs that we're going to do are going to kind of revolve around, not all of them, but one of them is going to revolve kind of around this topic of GMOs. And what's a GMO? Okay, yeah, so genetically modified organism. So when you take a look at genetically modifying an organism, basically you're taking them from what they naturally are and you're converting them into something that ultimately we want, right? We're kind of playing God. We're kind of playing the whole puppet maker, puppet master with these organisms. Somebody in the first class is like, well, we're designing them to live, in the environment or be more suitable for their environment. And like, that's not the case, like whatsoever. Plants, maybe. And the reason why I would say plants is because we're trying to produce like a plant that can maximize or has a better yield. That produces more, right? And best bang for their buck. And by doing that, something like Roundup Ready corn and Roundup Ready beans, well, what it is, you can spray Roundup on it, which naturally kills any plant out there. But in this case, they have changed the genetics behind these plants. So now you can spray Roundup on it, it kills everything else except for the plants. So I don't really disagree that we're making them more suitable for their environment. But when you think about it, we're not doing it for them. We're actually doing it for us, right? And now we kind of take away the whole process of natural selection. And now we're starting to kind of maybe play a little more with artificial selection, right? And then you kind of start throwing the whole ethics side to it. Well, is it ethical to actually take and do that with an organism, right? And you got plants, yeah, people like animals, no. Can we do that with humans? Can we genetically modify a human? Oh, absolutely we could. Yeah, no, I mean, it's ethical, right? Yeah, we should. There's guys out there that have tried it, but it's like we can't. So, and ultimately what genetically modifying is, you're basically going in, you're extracting genes to implement something else into it, right? We do it with bacteria all the time, insulin. Okay, we genetically modify them so that way they can produce insulin for us in a laboratory setting, right? And we can get mass production of it. Okay, we don't rely on just simply our body to produce it. We have things do it for us. And so when we look at these pictures, that's kind of the whole point about it. Is that the one on the right has been genetically modified, right? Same plant. Right? The one on the left has been getting destroyed by this little bollworm. Right? The one on the right, no impact whatsoever. I think it produced like a bitter tasting leaf that the bollworm, the worm didn't really want to even go into and bite. So again, when you look at this, we could play the whole ethics game. We could play the whole animals like, man, that's not right. It's like illegal. It's not ethical to do with humans. But then on the flip side, we're doing it with so many other organisms out there because again, it's helping us to hopefully survive, in the environment that we're in right now. So that's kind of the ultimate goal. But in order to get to this whole process of gene regulation and genetic modifying, we've got to understand, obviously the basics behind it, right? And so when we go into, we're going to talk, I'm not going to talk a lot about history. The one thing I want to bring up about history is that there are more people involved in the finding of DNA than just Crick and Watson, okay? And they're the two guys that generally get all of the credit, right? Now, the more and more that you dive into this, though, right, this lady right here was basically the one. She was a founder. She was the one that actually started the research, right, and actually did most of the work. She just happened to die. The other two picked up her research. Right, and expanded on it, and were able to really kind of concrete or solidify what the DNA molecule was. All right, for the most part, when they go through, it's like this is really all we have, really kind of understanding of the DNA molecule itself. Right, is this kind of crystalline shaped structure. Right, and so we're able to go in. Now we're able to go in and get a little bit better views of them. But starting at the beginning, like here's a top down view of basically this DNA strand, and that's all there was. right? And Franklin took it a step further, basically this discovery of it, and Watson and Crick took it a little bit further. They actually used research to figure it out, right? And so obviously they get all the credit. But good, just things that you need to kind of understand about the history is that it's not always Watson, it's not always Crick. You got to give credit to what we need credit for, okay, or who needs credit. Now, I think here's where we're going to go. So we're going to talk. Let's go this route. I'm going to start here. We're going to go with structure of DNA right off the bat, okay? And really what we're going to look at is we're going to look at the differences between DNA and an RNA structure, okay? So just to get the basics down, and this is basically what your whole packet was all about, right? Now, if we take a look at basically a nucleotide, right? Remember a DNA molecule, right, or an RNA molecule, we could also label these as a polymer, right? Okay, just like we did with carbs, like we did with lipids, just like we did with proteins, nucleic acids were also thrown on that whole polymer list. Huge macromolecule, right? Polymer, made up of individual parts, aka monomers, that we call, nucleotides, okay? So when we go through and take a look, we can take a look at this DNA structure as a whole, but then we can also break it down, obviously more specific. And that's kind of ultimate goal here is we're going to start breaking this down, to the actual chemical composition, right? The structures of these molecules that make up these DNA and RNA molecules themselves. So let's start here. DNA versus RNA, okay? If we're going to talk about DNA and RNA, right, they're both made up, first of all, of a nucleotide with a sugar, a phosphate, and a base. That's one thing that they have in common, okay? But there are some differences that we're going to find amongst them. Okay, one, and these pictures are actually backwards, and from how I wrote this out earlier, and just all based on this lettering here. So again, this would be a DNA strand over here. Okay, here's the RNA strand over here. Okay, so let's talk first of all strands. Let's talk big picture. Okay, when we take a look at the DNA molecule, it's double helix, twisted ladder, double-stranded. RNA, single-stranded molecule. Okay. If we take a look at the sugars that represent each of these molecules, in the case of DNA, you've got deoxyribose. Okay. So in the case of deoxyribose, remember OSE at the end means that it's a sugar. Yep. So the nucleic acid, meaning that it's a nucleic acid, right? Here's deoxyribose nucleic acid. And the RNA, you've got a ribose sugar. What's the difference? Deoxy? No. Here, we're not talking dioxy, we're talking deoxy. There's no oxygen. Okay, so when you look at the composition, it's all about the makeup. Just simply doesn't have an oxygen, right? And I can show you that structure here in a little bit, right? But that's all it is. The ribose has oxygen, deoxyribose doesn't. And based on that oxygen, that's where you're getting, again, the different shapes that we're seeing here and the different molecular, the compositions that make them up, right? Okay, phosphates are the same. Okay. Bases, a little bit different. Okay. And in the bases, we got ATCGs, adenine, thymine, cytosine, and guanine. And then you've got AUCGs, adenine, uracil, cytosine, and guanine over here. So the only difference that we're finding is that this T is a U in uracil. Okay. Now, we can go through and talk about, why they are the way they are. And I think a lot of it, again, has to do with the overall, the what makes up that ribose sugar versus that deoxyribose sugar, right? But then again, we can also kind of take a look at the evolutionary standpoint on why, DNA versus RNA. We'll address that a little bit more in the future because actually there's three types of RNA. There's an mRNA, tRNA, and rRNA. The one that's probably more common to us is going to be the mRNA, right? And that's kind of what we're looking at over here with this guy right here. But there are purposes, all right? And we'll get into that when we start talking about the overall protein synthesis and why it's important that this RNA basically is a single-stranded molecule. And it is also relatively, the one I'll throw in there, RNA, again, it's relatively unstable compared to DNA. And that's all relative because even DNA is not very stable. It's got weak hydrogen bonds that actually hold the nitrogen bases together. And those weak hydrogen bonds are weak for a reason because, again, it allows that molecule to separate and come back together and separate and come back together whenever replication is occurring, okay? Now, if we start breaking down, again, the overall structure, we're talking RNA versus DNA. We kind of start looking at the nucleotides. If we take a look at this as a twisted ladder, just remember you've got your sides and you've got your rungs. Okay, the sides basically are alternating sugar and phosphates. Again, in this case, we'd be looking at deoxyribose sugar and the phosphates. And then you've got the nitrogen bases that are basically stringing across, make up the AKA rungs of the ladder. and they're held together by hydrogen bonds. RNA, no hydrogen bonds. Why? Because there's not two sides. Yeah, there's just the one side, right? Okay, and that's what we're looking at there. Now, in the case of the overall nucleotide itself, okay, here would be a phosphate. Whoops, get out of there. Here would be our 5-sided sugar, and then here would be a nitrogen base. So this right here would be a nucleotide. Another phosphate, another sugar, and here's another nucleotide. Here's another nucleotide. There is another nucleotide. And we're talking specifically about an RNA molecule here because there's a uracil instead of...
Excuse me, could I have Aiden Johnson to the office with this thing?
Yep.
Thank you.
So we have, in this case, again, we're looking at, a little bit different. And it's really not that different when you look at the overall shape and the overall structure, okay? Now, the other part that they brought up in your guys' little packet was this kind of concept of these pyrimidines versus purines. Okay, and any pyrimidine, and we're talking specifically Cs, and we're talking thymines or uracils, okay, are a single ringed structure. Okay, if we're talking purines are a double ring structure, like this right here. Okay, now you guys also, in the case of the DNA molecule, you learned about Chargaff's rule. Okay, and Chargaff's rule states that only certain nitrogen bases will pair up with certain nitrogen bases. And what are they? Yeah, A's will bond with T's or U's, right? If we're talking RNA, okay, and C's are going to bond with G's, okay. Now, if you take a look, realizing that C is a single ring structure, G is a double ring structure, right? A is a double ring structure, T or U is going to be a single ring structure, okay. And there's a little bit of an importance to this, okay. Now, envision basically a molecule to where if this was a free for all. Okay, if it was a free-for-all, basically meaning that G's could go with C's, A's could go with T's, or A's could go with G's, it doesn't really matter. Basically, any combination could actually happen itself, right? One, we'd be starting to look at basically the overall structure as basically being very what I would consider to be unlinear, right? Because sometimes you're going to have one ring, you're going to have two rings come together, you're going to have maybe a single ring and a double ring that come together. And so what you're going to find is that basically that molecule is just going to kind of get all out of whack, right? It's not very linear. Okay. Now, the other thing that we start seeing with this is, again, just about the process of, mutations and the process of replication, right? It's a lot easier, more efficient that if we have sides that correspond to each other for the enzymes to come in and they simply can read those and they can just zip right down and be able to build two exact copies, right? If it was basically a free for all for what organelles or what molecules this could attach to what, there really wouldn't be a plan as far as going in and doubling up or replicating the molecule. Do we understand that? Yeah, go ahead. What are rings? So it's a shape. So here is that's a ring. Okay, over here would be, this would be a double ring. Yep. So again, when you look at it, again, a lot of it has to do with the actual structure, but two, just the replication process itself, it wouldn't be anywhere near as efficient as it is right now because we wouldn't have and again, we'll talk about these a bit further. We wouldn't have these molecules that could just, we got an A. Well, I got to bond up a T. Oh, there's a C. I got to bond up a G. Instead, it'd be like A. It's like, man, I have no idea what goes together because it could be a T, it could be a C, it could be an A. I don't know. And how would it know? Okay. So much more efficient in this process when, we start looking at these molecules being able to stick together. Okay. So DNA versus RNA. Right, going to be very, very important when we start looking at it. Obviously, the DNA that makes up, or the nucleic acids that make up our cells can be DNA, but RNA has a very, very special place, right, when we start looking at protein synthesis, right, during the process of transcription and translation. And we'll explain that a little bit more in the future. Okay, any questions on the overall structure? No. Okay, now, let's go here. So here's the other part. Okay, based on the structure, this is going to be very important to you guys during replication. And when we look at these molecules, and this is kind of where we start to get a little bit more further in depth, right, in the overall molecule itself and how the bonding simply occurs between the two sides, okay? So when I take a look, okay, first of all, we call these anti-parallel strands, okay? And anti-parallel simply means that they are running side by side, but we got one that's running in one direction and the other is running in the other direction. The question is, how do you know which one is running which direction, okay? And the reason why this is going to be important because when we start breaking down replication, remember replication is all about copying DNA and ensuring that when a cell divides, it has the exact same copy of DNA. That can only happen in one order. That can only happen from one end to the next. And so it's important that we know, again, which of these basically are, the top, so-called top or so-called bottom, okay? So here's where we're going to start to go, okay, or the terms that we're going to start to use when we start explaining these corresponding strands is we're going to start using terms called 5' and 3', okay? Now, you're going to have a 5' end to every side, and you're going to have a 3' end to every side. And this is all based on, right, the direction or which way the sugar is facing or sitting, okay? So if I take a look at this sugar right here, if I take a look at it as a pentagon, I would consider that pentagon to be sitting upright. Okay, to me, that would be an upright pentagon. Okay, now if I go to the opposite or the anti-parallel strand or the corresponding strand, what do we notice about that pentagon? Yeah, I would say it's upside down. Okay, so when we take a look, all of these pentagons are actually, to me, I'm thinking, oh man, they're running like upwards, right? Because that's how, I'm seeing that the tops are up here and the bottoms are down here. And if I take a look at this side over here, to me, I'm thinking, well, they're running from top down because my tops are down here, okay? Now, what really kind of refers to, or how you guys need to refer to this, is in the terms of these 5' versus 3' ends, okay? And so here's what we're going to do. So there's a certain carbon that you're going to find. There's actually 5 carbons that are found on these sugars, okay? So in this case, I've numbered them off for you. There is going to be a carbon sitting right there. Number 1, they number this one. Number 2, number 3 is right there. Number 4 is right on that point. And then there's another carbon that extends out from that 4th carbon. Okay. So when you read, right, this molecule, your 5 or 5th carbon is up on top. So that is your 5' end. Okay. If I look at where my 3' end is at, or my 3' or my 3rd carbon, where's that at? It's on the bottom, right? So this would be my 3' end. Okay. If we jump to the other side, okay, what end am I looking at right here? 5' or 3'? Yeah. 1, I know that because it's anti-parallel to this side over here, right? Okay, I know that it's anti-parallel to this side. Where's my little mark? There it is. Okay. But also by looking at that, where's that 5th carbon located? That 5th carbon is located down here, and this 3rd carbon is located up here. So again, this would be my 3' end. Okay? And I'm going to stop there as far as the structure goes. There's going to be much more when we start looking at the whole process of replication and why it matters. But again, simply being able to acknowledge where that 5' versus that 3' end is at on these DNA molecules. Okay? Questions there? You guys are good. Okay, let's go back up because now I want to kind of hit this out a little bit. Since we've talked about DNA a little bit versus RNA, now let's talk about a little bit about eukaryotic versus prokaryotic organisms. Okay, and the ultimate goal is that we kind of want to be able to go through and talk about some of the similarities, but also talk about the differences. Okay, now realizing if we're talking bacteria, we're talking prokaryotic organisms, eukaryotic, they're both made of DNA. Okay, the actual nucleotide itself, there's nothing different. Sugar, phosphate, deoxyribose sugar, phosphates. Right, and then you've also got the nitrogen bases, A, T, C's, and G's that basically span across. There's nothing different there. Okay, so for you guys, where do we start seeing? Because you've done this in the packet that we did on Friday and on Monday. Okay, where are some of the differences amongst the prokaryotic versus eukaryotic organisms in terms of DNA? In what? You are eukaryotic. You have a nucleus, right? Yeah. So 1, right? If I start looking at prokaryotic, I'm just going to write this out over here, versus eukaryotic, right? We can talk about, nucleus. Prokaryotic organisms don't. Okay. And we have addressed this in the past, and I'll come back to you. Okay. We've addressed in the past. Always a big question is why? If eukaryotic organisms have them, why don't prokaryotic organisms have them? Or if prokaryotic organisms were the first organisms to be found on Earth and it works for them, why doesn't it work for us to not have a nucleus? And that was a really good discussion today for our 3rd period class. And I'll come back to you on your second, whatever you got going on there, but let's talk about that first. Why is it important? Why is it important that we have a nucleus? What do you got?
Easier to protect it and it's hard to mutate.
Okay, 1, Easier to protect. Why is it easier to protect? Because we have a what surrounding it? Yeah, we have a membrane. Now, the other part that we need to understand, we've talked enough about membranes, and we've talked enough about, well, we've got this endoplasmic reticulum, and we've got mitochondria, we've got chloroplasts we find in eukaryotic cells, and they all have their own membrane. Okay? So, yes, that membrane is there for protection, but what else does that membrane do? Yeah, it provides its own environment. Very good. And so you may even find specific enzymes that are embedded within that membrane or within that membrane, or I should say within the organelle itself, that will aid in whatever that organelle needs to do, right? So in this case, if we're taking a look at a nucleus, okay, what kind of enzymes would you expect to find? I'm losing, McKinley. You're going down hardcore. Okay. Yeah, you're getting heavy eyes. Okay, so if we start looking at what kind of enzymes would you expect to find in the nucleus? Okay, ones that would help with replication. Very good. Okay, maybe ones that would actually help to build an RNA molecule, right? Okay, some of those. That again, if you don't have a nucleus, where are those enzymes located? Oh, they'd be there, but they'd be anywhere, exactly. And when you take a look at a eukaryotic organism, there's a lot of crap going on in our cells, right? To where all of a sudden now we start looking at the inefficiency that if I've got enzymes that are over here, but my DNA is over here, it's got to travel and it's got to be able to get to it and figure out where it's got to go. Okay, which is probably not such a big deal for prokaryotic organisms. Why? Yeah, they're small. They're not as complex. Very good. They're not as specific in what they do. And even then, taking a look at their DNA, right, which is kind of number 2, right, their DNA is relatively small. It's relatively little, right, as far as it goes. And it's also, and it's also just in a single loop. Now, in the eukaryotic organism, we go, I'm going to put relatively large, okay, and it's relative. I'm going to say small over here because, again, we interpret eukaryotic organisms as being more complex. If we're more complex and more specialized, we're going to have more genes, okay, which obviously can control our body, more things going on, right? Now, the other part that we start looking at here is it's in a single loop. I still haven't really found an advantage to this yet, okay, other than when you start looking at the chromosome, and I say chromosome because most bacteria have just one, okay, where ours are obviously linear. Now, they're not always that way, okay? Like when I scroll through or to this slide right here, for the most part, we've got what they call chromatin. And chromatin is basically this DNA molecule that's just kind of just inside a cell. And if you remember, like if here is a cell in interphase and it's got the nucleus, right? You can't see specific chromosomes in that nucleus. It's all just strung out, okay? It's not until they start to go through the process of mitosis to where now you start to see the actual chromosomes that we would interpret, that we always think of when we think of chromosomes. And there's a reason behind that as well. We'll talk about more gene regulation. But when we think about gene regulation, the ability to turn a gene on and off, right, is happening continuously inside of our body. Well, why would you think that would be a problem to turn genes on and off if it is in the form of this chromosome versus just kind of being strung out like we see most of the time during interphase? Harder to find. Because again, it's wrapped around these histones, right? And then it's coiled up even more. And so there's genes that could be sitting like inside of the actual chromosome itself, right, that are tougher to get to, right? And so again, they want to spend most of their time in this case because it's a lot easier to, again, turn genes on and off and go through some of these processes, right? So again, we start looking at that shape of that linear approach versus that loop. Obviously, we have many chromosomes. There's got to be a breaking point somewhere, right? Okay, I mean, it's just not one continuous strand of 46 chromosomes in this case, right? That is easy for them. Prokaryotic also have plasmids, and we'll deal with this a little bit more when we start to get into like our pGlow gene lab. And the plasmid is simply just a secondary little loop of DNA that we find down here. And now what researchers are starting to find is that this is kind of the DNA that they can exchange genetic information with other bacteria. So if you've got one that's like antibiotic resistant, it can actually transfer that resistance onto another bacterial cell, which can then transfer that onto another one until they all become eventually antibiotic resistance, right? Basically kind of changing their DNA up just a little bit, okay? So do you have any more? I kind of jumped ahead of you. I apologize.
I was going to say junk DNA.
Yeah. Thank you. Okay, so where do we find junk DNA at? Where don't we? We've got junk DNA. What do we call those? Introns or exons? Introns was the junk DNA. Exons was the good stuff, okay? and there's no junk DNA over here. So now we kind of start looking at, well, what's the purpose? Why do we want junk DNA in an already complex system in the first place? Okay, why is it important that we have all this excess amount of DNA? Wouldn't it just be simpler to have just straight up DNA that we need? Wouldn't it make things much more efficient?
It's on the ends of the mutations. So
Yeah, one, I kind of like that. They eat the mutations, right? That's kind of a good way. Yeah, when you kind of think about it, when mutations happen, if all we have is just our regular strand of DNA and no junk, mutations can impact us somehow. May not always mean a different protein, may not always mean a negative or positive effect, but somehow it's going to change that DNA strand, right? But now if we have junk DNA built in and a mutation happens primarily to that junk DNA, does it impact us? No. Yeah. Really nothing happens. Okay. And the other part that we start to see, and we'll talk about this in the case of transcription, is that on the ends of these DNA strands, you're also going to find like these little, like they're called poly A tails and methyl caps that you're going to have kind of on the ends of these DNA strands. And I always kind of like to refer this to like a whip, for example. Right now, one, we have like these destructive enzymes that we have inside of our system. And the reason why they're there is because when we make something like mRNA and it goes to the ribosome and it forms a protein, we don't want that mRNA just to sit around, because if that gene gets turned off and that mRNA is still there and a ribosome latches onto it, what happens? It just keeps making more and more and more. So every time an mRNA strand is made and it goes through the ribosome, it's very unstable, which means that it can break down easy. We've got these enzymes that will actually take it and break it down. Okay. So one of the other kind of, junk DNAs are in the form of these methyl caps or these end caps. And I kind of refer to like a whip, right? If you're there cracking a whip, right, what's generally going to be the first thing to fray off and eventually get busted and broken down? The end is, right? Okay. And these destructive enzymes are kind of working in the same way, not like a whip, but they're going to focus on the end pieces first. And so if they can start to break those down, they'll never actually reach the DNA on the inside, and therefore that process can go through and happen. So, we always think, well, the enzymes must be bad. Well, no, they're good because they've got to break that stuff down eventually. It's just we don't want it broken down early on. We want to make sure that it's after the case is done. Okay? I think that's about it, right?
So I had an idea on your loop question.
What do you got?
You think it can easily replicate the loop and just duplicate a strand next to it? And then if it loops back around, it can pinch off that side. That's how it can get that excess.
Yeah, I know I do. And I think a lot of this also just, I guess probably not. My rationale is not all that great because, again, why wouldn't it just be simply linear? It could be just one linear chromosome when you think about it. But, yeah. I don't know what it is. I don't know if there's even a hypothesis out there as far as what it is, but obviously there's got to be a reason behind it and why it's that much easier. But what it is, I don't really know. Okay, yes, ma'am.
Why do the bad enzymes or mutations happen from like the sides? Why can't they start like in the middle?
Well, they probably could. I'm just saying it's most of the time, is it going to be easier to access? The ends are easier to access the middle. Like if you're eating a sandwich, for example, do you attack the middle of it? You do? Really? So, like, I don't know. Just to me, it's always easier to attack the ends, right? So, that's just.
Then the meat doesn't go shooting at the end.
Yeah, there you go. Yeah, because if you bite down the middle, then the meat just goes out both ways. We don't want that to happen, right? Yeah. But, that's probably my best picture of it. It just simply, I don't know if I really have a rationale behind it other than it's just probably easier to access those ends and work in from there.
So the DNA also